to discuss exploring the biological sulfur oxidation pathway using genetic engineering. Solid-state sulfur is a mineral which is abundant in the environment, and it can be found in a lot of oxidized and reduced states. Microbial activity is very important for cycling in between these oxidized and reduced states of sulfur in the environment. But when it comes to the interactions that microbes have with elemental sulfur, little is known about it, and there's a lot of gaps in our knowledge. The SoxC pathway is an enzymatic pathway used to describe sulfur oxidation, and the SoxC pathway is used by a variety of microbes. The sulfur oxidation process is the process by which electrons are given up by sulfur molecules, but the functions of SoxC and SoxD genes within the SoxC pathway are not well understood, and the project examines to look at what the functionality of these genes are. It has been hypothesized that SoxC and SoxD help with extracellular electron uptake. When we consider a model system for understanding the sulfur oxidation pathway, Thioclava electrotropha is a bacteria that can be used for this. Thioclava is a short-rod bacteria that is found in a lot of marine environments, and it has the capability of uptaking electrons from the extracellular environment. Now, Thioclava contains the SoxC and SoxD gene, which encode for proteins that are responsible for sulfur oxidation, specifically oxidation of thiosulfate to tetrathionate. And because of this, Thioclava is a good candidate for this project. So the big question that we try to answer is, does Thioclava use SoxCD for elemental sulfur oxidation? Through genetic engineering, our goal was to remove the SoxCD gene, and the techniques that we used for the process was conjugation and homologous recombination. Conjugation is the process by which genetic material is transferred in between bacterial cells, and homologous recombination is the exchange of genetic material between two similar or identical DNA molecules. Using these techniques, we hope to create a Thioclava mutant which lacks the SoxCD gene, and this will be referred to as a knockout mutant. The SoxCD knockout mutant will then be evaluated for the ability to oxidize inorganic sulfur compounds by plating them on inorganic sulfur plates. If the bacterial strains are no longer able to oxidize sulfur, then that could potentially show that SoxCD is responsible or helps with extracellular electron uptake. Future experiments will focus on the potential role of SoxCD in extracellular electron transfer with an electrode. Understanding the mechanism behind inorganic sulfur oxidation in Thioclava will help researchers to better understand the extracellular electron transfer mechanisms. And finally, I want to thank the Row Lab at the University of Cincinnati for making this project possible.